Thanks so much for the opportunity to present at the PacBio Neuroscience Day 2020 in this virtual format. I'm excited to share with you our clinical genetic study of a Mexican kindred with an ATXN10 repeat expansion and tell you more about how optical mapping and Cas9 smart sequencing supports our understanding of clinical molecular genetics of repeat expansion disorders. Spinocerebellar ataxias are inherited in an autosomal dominant manner. They are a highly heterogeneous group of disorders with a complex genotype-phenotype spectrum. They present with a progressive degeneration of the cerebellum, but can also affect other brain regions, including the brainstem. The core triad of symptoms are gait ataxia and incoordination, nystagmus and visual problems, and dysarthria. The types are described as SCA, followed by the number in order of their identification. Overall, the global prevalence of SCA is 3 in 100,000, but as you'll see, there is a wide regional variation. Treatment is only symptomatic, and currently there are no disease-modifying therapies available. Now, more specifically, the disease I'm talking about, the spinocerebellar ataxia type 10, is a rare autosomal dominant disorder. It's presenting first with gait ataxia, followed by dysarthria, nystagmus, ocular dyskinesia, and some patients also develop dementia or other mood abnormalities. In addition, a lot of people show seizures that are a common feature and usually occur several years after the onset of the initial symptoms. The average onset is 34 years, and the average disease duration is about 13 years. And also here, there are no available therapies except symptomatic anti-seizure medication for the control of seizures. The distribution, oops, I'm sorry. Interestingly, Cas10 cases have been primarily found in Latin American countries and are basically non-existent outside the Latin American populations, except for single cases in the US, in China, and Japan. These observations, combined with the relatively wide geographic distribution of SCA10 throughout Latin American countries, have led to the hypothesis of a founder mutation that likely arose in the ancestral Native American population when migrated from East Central Asia across the Bering Bridge to North America and then spread throughout the Americas from the north to the south. The map on the right is from a recent review that shows the prevalence for autosomal dominant SCAs in North and South America. You see the percentage of SCA of the total SCA population is between 5 to up to 45%, whereas it's completely absent in the US and Canada. The pathological hallmark of SCAs are GABAergic Purkinje cell loss and cerebellar atrophy. This is illustrated in this MRI and circled in yellow in the upper left panel. Below, you find an MRI from a SCA10 Parkinson's case. In panel AB, you see mild atrophy of the fovea indicated by the yellow arrows. C is a coronal section and shows expansion of CSF containing spaces. And in D, in the sagittal T1 view, you see mild superior cerebellar volume loss. On the right panel, you see the neuropathological features in SCA10, which is predominantly the Purkinje cell loss as a primary pathological finding. Panel A, the H&E staining, reveals extensive Purkinje cell loss with a reduction in volume in the molecular layer. Panel C and E show calbindin staining, which confirms the loss of Purkinje cells. The remaining Purkinje cells are dysmorphic with markedly reduced dendrites in the molecular layer. And additional stains you see in G through I of glutamate decarboxylase, which is the rate-limiting enzyme that catalyzes the conversion of glutamate to GABA, further confirms Purkinje cell loss in SCA10 cerebellar cortex. Now, the genetics of SCA10 is based on an expansion of an intronic pentanucleotide repeat in the ATXN10 gene, which was first described by Dr. Ashizawa's group in 2000. The normal repeat length is 10 to 32 repeats. Intermediate repeats are 280 to 850 repeats. And we see in general full penetrance of the disease over 850 repeats. And of note, repeat alleles between 33 and 279 alleles have not been reported to date. Furthermore, there is an ancestral common haplotype that has been identified and that is shared in all families, which again strongly suggests a common founder. This is the original pedigree from our family. In black, half black, and gray symbols are indicated affected cases. The proband here in generation 3.8 originally presented in our clinic with Parkinson's disease about 15 years ago. And the SCA diagnostic panel for him and his brother showed an ATXN10 expansion. We were puzzled at that time about the clinical presentation of Parkinson's disease and SCA10 in the same family. But at that time, this was all we could do with the technologies of PCR and Southern blot that was available. As you know, Southern blot analysis can only roughly estimate the repeat length and size of a DNA fragment containing the repeat expansion. And while the more recent developed repeat prime PCR on the right panel that relies on the annealing of a repeat primer to the expansion allele and produces a regular 5 base pair ladder of the PCR products, it cannot resolve the complete repeat expansion of ATXN10, but only about 1 to 1.5 kilobases at the 5' end, and not the several kilobases that repeat extends. So the exact size and repeat composition cannot be detected with these techniques. Fortunately, technology has advanced, and PacBio No Amp sequencing and BioNano optical mapping can be very powerful to solve the genetics of these long repeat expansions. We wanted to analyze DNA samples from affected and unaffected family members of the SCA10 family and apply Cas9 smart sequencing from PacBio and optical mapping using BioNano genomics. We wanted to do this to understand the phenotype-genotype correlations. For example, how does the repeat length affect the clinical phenotype? How does repeat composition affects clinical phenotype? And possibly, can we assess somatic mosaicism of the repeat expansion? For this study, we recruited additional family members of the 4th generation to be able to study the expansion in additional cases and also really assess intergenerational changes. We first used high-resolution optical mapping, which is a next-generation cytogenetics technology. In principle, this technology relies on fluorescently labeled ultra-high molecular weight DNA, which is run on a flow cell with nanochannels and then imaged. And the images are analyzed and assembled to a genomic map to detect the structural variants. You can also find more information about the technology on the BioNano website. So here are 2 examples that we analyzed. In this case, it's a parent-child duo. We analyzed by optical mapping, and you see individuals of the generation 3, 4, and he has a 6.2 expanded allele, and his daughter, 4.7, carries an expansion of up to 13 kilobases with a number of smaller expansions indicated by the distribution of the green signal in the histogram below. The distribution of these alleles are also shown in the box plot on the right. And keep that individual in mind. You'll see that later in the presentation. And then the second technology we applied was the PacBio No Amp targeted sequencing, with the goal not only to resolve the repeat size, but also the repeat composition. The workflow for No Amp sequencing starts with preparing the SMRT libraries using high-quality genomic DNA, then block the 5' and 3' ends to prevent off-target ligation, use custom-designed guide RNAs and Cas9 digestion to enrich for the region of interest, multiplex samples, and then remove off-target products before you do the sequencing and data analysis. To explore the repeat structure, there are a couple of different ways to display the data. So one is the waterfall plots, which outlines the repeat structure of the 2 alleles. On the y-axis, it shows the reads, and on the x-axis, it shows the length of the repeat expansion in base pairs. And then on the far right, you can also use histograms to examine repeat characteristics, and then the repeat counting allows for the confirmation of the allele sizes, both for normal and expanded alleles, and shows the extent of mosaicism of the expanded allele. So here are examples that we did in this family, and Cas9 smart sequencing confirms the sizing of the optical mapping quite nicely. In the past, the Southern blot analysis estimated much larger sizes of the expansions, but importantly, No Amp sequencing really fully allows us to resolve the repeat composition, and that's what's shown here. In the upper right corner, I highlighted a partial pedigree so that you can orient yourself which samples we were sequencing. And on the panels, you see samples 4, 5, and 6 that have a biphasic pattern of the repeat and show some mosaicism. And you see the ATTCT repeats represented in gray, and ATTCC repeats represented in orange. And these are both from unaffected individuals from a case, which is 3.3, which has been affected with SCA10. and also has seizures. And in the middle, you see samples 3.4, 4.7, which we've also done BioNano mapping for, and also 4.8. And they show a different pattern. They show a pure expansion. And it's also interesting in 4.7, you see a very large variability of the repeat length. And when we are measuring these repeat length, we see repeats between 66 and 2,782 ATTCT repeats. So none of them are affected, although the ATTCT repeat counts are over 850 repeats. So for the 3.4 case in the middle, we counted 950 to 1,435 repeats. And this individual is 70 years old, but clinically unaffected. So it seems like there is also a range in penetrance for these different repeats, ATT, CT. So I only showed you some examples of the data, but I indicated here with asterisks samples that were analyzed with PacBio, and indicated with hashtag samples that we analyzed with the BioNano Optical mapping. The black symbols again indicate the biphasic repeat that resulted only in the SMRT phenotype, and in this case also with seizures after the age at onset. And in red, the red symbols are cases with the pure repeat expansion. And really importantly, in the third generation, two of the individuals are currently at age 65 and 70 and are not affected yet, even though they carry more than 850 ATTCT repeats and should be symptomatic. And then one case that is highlighted in the complete red square we published in the past has the pure repeat and shows typical signs and symptoms of Parkinson's disease. So in summary, I could show you that the repeat length and detection of mosaicism is comparable with both technologies, BioNano and also PacBio. And in addition, PacBio identified ATXN10 expansion structure, and we found a mixed repeat structure of ATTCT repeats together with ATTCC repeats in 10 family members, and also the pure ATTCT expansion in 6 family members. And interestingly here, 2 have not been affected to date. We found intergenerational repeat expansions, both detected through maternal and paternal transmission. And the mosaicism of the repeat expansion was independent of the source of the DNA. So we have both saliva samples or blood for these individuals. So in the last couple of slides, I want to go a bit into the pathophysiology and show you how these repeat expansions look like at the cellular level. And as a background, there are several molecular disease mechanisms described for repeat expansion disorders. Pathogenic coding glutamine expansion repeat length leads to translation of expanded abnormal polyglutamine repeats, which lead to protein misfolding, cell toxicity, and neurodegeneration. For SMRT10, we look at non-coding repeat expansions on the right, which leads to toxic RNA gain of function. And the hallmarks here are RNA foci consisting of transcribed nuclear accumulation of expanded RNA with repeat RNA binding proteins that then can cause RNA toxicity, and cell death. So what we've done here is we used a custom-designed RNA-scope hybridization probe to the ATXN10 repeat. What you see in panel A through C is a positive control of human RNA. In the middle, panels D through F shows a healthy control skin fibroblast line without an ATXN10 repeat expansion as a negative control. So in E, you see a black panel. And then in G through I, we have skin fibroblasts from an ataxia-10 ATTCT repeat expansion carrier with highly variable repeat length. And you've seen the results from the PacBio sequencing before. We counted 66 to 2,800 ATTCT repeats. When you look at the panel H and I, you can count the number of DAPI positive cells, and those are 11 in total. And of those, 7 of them show a positive signal for the ATTCT probe, whereas 4 of them are negative that you see that are indicated with the red arrows in panel I. So presumably the long expansions lead to prominent aggregates in the cells, whereas the shorter repeats that this individual also carries show only small puncta or no aggregation of the repeat RNA at all. So in conclusion, CRISPR-Cas9 single molecule real-time sequencing technology combined with optical mapping, cytogenetics really allows for accurate sizing of the repeat expansion, resolves the entire repeat composition, and establishes somatic mosaicism. This allows us for exact repeat sizing and composition, and this is really critical for clinical practice and genetic counseling because we can discuss disease penetrance, which might not be fully penetrant for some of the compositions, the clinical symptoms. We know that seizures, for example, relate to the ATTCC repeats. And we can consequently also discuss the disease trajectory with our patients. In the future, we want to develop these panels as multiplex panel with Cas9 sequencing, and we are also interested in studying further underlying molecular disease mechanisms. Last but not least, I want to acknowledge my collaborators, and I'm very, very grateful to the family participating in the research. In particular, I want to highlight Dr. Javier Jimenez, our neurologist from Guadalajara, members of the Schuele lab, Joss Palafox, Faria Zafar, Ali Torres. From PacBio, I've been very grateful to work with Yuji Zai and Janet Ziegler. And the BioNano team is composed of Carl Hong, Joyce Lee, Jill Lay, and last but not least, our long-standing collaborator and SCA expert, T. Ashizawa. Thank you for watching. And if you have comments or questions, please contact me, follow us on Twitter, or visit our website. Thank you so much.